Good morning, students. The last topic of biological oxidation is and computation number 6.6 is shuttle system for transport of extra mitochondrial NADH. In the previous lecture, what we have studied? We have studied that NAD is present in the mitochondria. Various substrates will donate their hydrogen atom to NAD and NAD is converted into NADH. That NADH then donate its electron to complex 1. So all this occurs in mitochondria, but there are some cycles like glycolysis where NAD is converted into NADH in the cytoplasm. Now when this NADH is synthesized in the cytoplasm, there is no electron transport chain in the cytoplasm. This NADH has to enter into the mitochondria to donate its hydrogen atom or electron to the electron transport chain. But inner mitochondrial membrane does not allow this NADH to travel through it. Hence, two shuttle systems are present for transfer of this NADH from cytoplasma to mitochondria. First shuttle is called as malate aspartate shuttle. It occurs in majority of the tissues. And second shuttle is glycerol 3-phosphate shuttle. It mainly occurs in muscles and brain. So this is the first shuttle, malate aspartate shuttle. The central dark band is representing the inner mitochondrial membrane. On the left side, cytoplasma is present. On the right side, mitochondria is present. So the NADH is synthesized on the cytoplasma. Now this NADH, we have to transfer this NADH to the mitochondria. In the mitochondria, this NADH will donate its electron to the electron transport chain and ATP can be synthesized. Now what happens here? NADH, as I have told you, is impermeable by inner mitochondrial membrane, therefore it does not transfer directly. Therefore, NADH donate its 2 electron to oxaloacetate, and oxaloacetate is converted into malate. The enzyme is malate dehydrogenase. Now this malate is permeable through the inner mitochondrial membrane, and malate, when concentration of malate in the cytoplasma increases, it travels across the inner mitochondrial membrane and enter into the mitochondria. In the mitochondria, the reverse reaction occurs and malate get converted into oxaloacetate in the presence of enzyme malate dehydrogenase. In this process, 2 hydrogen atoms from malate are removed and added to NAD+, and this NAD+ is converted into NADH. So in this way, the NADH is synthesized. Now oxaloacetate is formed during this process. This oxaloacetate, again we have to transport to the cytoplasmic compartment. But again the problem is oxaloacetate is impermeable by inner mitochondrial membrane. Therefore, this oxaloacetate undergoes transamination reaction. Here, in this transamination reaction, the amino group from glutamic acid is transferred to oxaloacetate, and oxaloacetate is converted into a amino acid called as aspartate. During this process, glutamic acid is converted into alpha ketoglutarate. The enzyme required for this reaction is aspartate transaminase present in the mitochondria, AST. Now aspartate is permeable by inner mitochondrial membrane. Aspartate enters into the cytoplasma, and again the reverse reaction takes place. Amino group from aspartate is removed and added to alpha ketoglutarate, which is present on the cytoplasmic compartment, and this alpha ketoglutarate, after addition of amino group, get converted into glutamic acid. Again the enzyme is same, aspartate transaminase. So in this way, this shuttle starts from oxaloacetate and ends with oxaloacetate. This shuttle takes place in various tissues. The next shuttle is glycerol 3-phosphate shuttle. In this shuttle, as I have told you, it occurs in brain and muscles. Again, central line dark band shows inner mitochondrial membrane, left side is cytoplasmic compartment, right side is mitochondrial compartment. Now again NADH is synthesized on the cytoplasmic side. This NADH donate its 2 hydrogen atom to dihydroxyacetone phosphate. So this starts with dihydroxyacetone phosphate. After addition of 2 electron or proton, this dihydroxyacetone phosphate is reduced to glycerol 3-phosphate. The enzyme is dihydroxyacetone phosphate reductase. As the substrate is reduced, it is called as reductase. Now this glycerol 3-phosphate easily travels through the inner mitochondrial membrane, enter into the mitochondria. After entering into the mitochondria, this glycerol 3-phosphate is oxidized by donating the 2 hydrogen atom to FAD and to synthesize FADH2. FAD is reduced to FADH2 by accepting the 2 electron. And this glycerol 3-phosphate is converted into dihydroxyacetone phosphate. The enzyme is same, dihydroxyacetone phosphate reductase. Now this dihydroxyacetone phosphate reductase is permeable by inner mitochondrial membrane, it enters to the cytoplasm. So in this way this shuttle takes place. One point you have to remember in this shuttle is, see here NADH donate 2 hydrogen atom to dihydroxyacetone phosphate on cytoplasmic side to synthesize glycerol 3-phosphate. On the mitochondrial side, glycerol 3-phosphate donate 2 hydrogen to FAD and not to NAD+. Therefore, the product is FADH2 and not NADH. So, what is the difference here? If NADH is generated, it gives 2.5 ATPs, but here FADH2 is generated, so it gives only 1.5 ATPs. So one NADH generated in the cytoplasm of muscles and brain, after this shuttle it generates FADH2 which gives only 1.5 ATPs. This is about the shuttle system for transport of extra mitochondrial NADH. Thank you very much, and this complete our topic of biological oxidation.